Okay, let's go.
All right, hello everyone. Today, Karen and I are going to be presenting this paper titled Multimodal Contrastive Learning for Enhanced Explainability in Pediatric Brain Tumor Molecular Diagnosis. So let's kind of start with the challenge that this paper presents, which is that CNNs show great promise for brain tumor diagnosis from MRI images, but are not widely trusted by radiologists due to their lack of explainability, which hinders their adoption in clinical settings. Explainability here is specifically defined as the ability of a CNN to diagnose similarly to radiologists and attend to appropriate parts of an image, the same ones that radiologists would attend to. So what can be done about it? Well, why don't we integrate different sources of information derived from radiologists into...
Why is that requirement on the same things that radiologists attend to instead of just the high accuracy?
It didn't necessarily go into that. It just said that that's one of the reasons they don't trust it. And later on, they show, for example, that the model where the attention of the model is focused isn't on where the tumor region is. And there's just a lack of trust in whether they're going to perform well by radiologists.
Maybe what they think about attention is not what it is. That could also explain why. Okay.
So yes, well, one possible solution to this is integrating different sources of information derived from radiologists themselves into the CNN framework. Now, this has already been done in previous works with semantic features and eye gaze information. Eye gaze information refers to the parts of an image that a radiologist looks at for making a diagnosis, and it's usually collected by an eye tracking system. This can, for example, see, okay, the radiologist is focusing on this part of the image for this amount of time, and then their eye is tracking to this part of the image, and then going back and forth between these, kind of that sort of information. And semantic features are important features that are annotated by radiologists corresponding to a certain disease. But the challenge is that getting these kinds of data are expensive and labor-intensive, which makes them really hard to get. However, a different resource that is widely available is radiology reports, and they are most accessible in most medical imaging datasets that are out there. The other benefit is that they include some of the relevant information that they hope to extract from the previous two data sources. So the goal of the paper is to use contrastive learning to leverage radiology reports into a CNN-based framework for improving model explainability and also improving performance and downstream classification tasks. Let's quickly look at the contributions of the paper. There are three different categories. One is training a deep MRI report contrastive learning framework for learning generalizable MRI representations. The second is incorporating tumor location into the CL framework to enhance the representation learning process. And the third is fine-tuning this CL-based representations on the PLGG genetic marker classification to both improve explainability and performance. Pediatric low-grade glioma, or PLGG, is one of the most common brain tumors in children. I think it's 30 to 40% of the cases. And identifying its genetic markers are highly important for treatment planning, prognosis, things like that. But the major clinical method for identifying tumors is brain biopsy, which is invasive, difficult, costly, especially in children. And so there have been CNN methods that identify these markers, but they rely on regions of interest in the image, which requires additional masks to come with the data set, which is another step that needs to be taken. So they hope to bypass this need for regions of interest by the framework that they create. Okay, so I'll pass it on to Karen to talk a little bit about what contrastive learning is.
So contrastive learning is a type of self-supervised learning. And what self-supervised learning is, it's a type of machine learning technique where a model will learn from unlabeled data and either generate like labels or supervisory signals from that input. And so in contrastive learning, what this type of self-supervised learning does is it actually like finds relationships between different data points and determines whether they're similar or dissimilar. And so in the latent space, contrastive learning takes similar data and tries to push them closer together, and take dissimilar data and push them farther apart. And one of the ways contrastive learning does this is by using one of two common loss functions. And the first one is contrastive loss, where the model is, say, it's given two inputs, and let's say they're A and B, and if A and B are similar, it will minimize the distance between them in the embedded space or latent space. And if they are dissimilar, it'll make the distance between those two data points farther apart. And it does this with the loss function shown below, where if the distance between A and B is supposed to be minimized because those 2 points are similar, the y value in that function will take on 1, and that will cause the part for the dissimilar data shown in red to equal 0, and for the part that's shown in green, that distance between the similar data to have a value over greater than 1 that's positive. But what the loss function does is try to minimize that. So like the loss function will have a smaller positive value for the positive pair to minimize that distance. But for the negative pair, what it will do is it will try to maximize that distance in the latent space. And the other type of loss function is triplet loss, where the model takes in 3 data points. It will have an anchor, which is the first image, and then it will have another data point, which is supposed to be similar to the first image, and that will be like the positive data point, as well as the negative data point that is dissimilar and should be farther apart. So in the latent space, there is that blue line called the margin. And so what the model tries to do is it will take that green positive data point and try to make sure that the red negative data point is both farther than the margin and the distance between the anchor and the positive pair. And so what that looks like in the loss function is the loss function will try to maximize the distance between the anchor and the negative data point, and it makes sure that it's both larger than the margin distance and the distance between the positive pair and the anchor point. And so in this sense, the positive will always be closer to the anchor and the negative data point will always be farther.
Okay, so with that background information, let's get into how the paper accomplishes their goals that they set out, and let's specifically start with the data sets used. The study uses 2 internal data sets called the SIGKIDS data sets. Dataset 1 has 341 3D FLAIR scans from 341 unique patients. Those come with corresponding radiology reports, manual tumor segmentation masks, and genetic marker labels. It also comes with tumor location information, which is represented by 3 different classes specifying the location of the tumor in the brain. Those 3 are right there. I'm not going to try pronouncing them. And 204 of these samples are associated with BRAF fusion and BRAF V600E mutation, which are the 2 mutations for the PLGG tumor. And these 204 data points are going to be used later on for fine-tuning the CL model on the downstream classification tasks. And dataset 1 is the training and testing dataset, and dataset 2 is the validation dataset, external validation dataset. That one has 76 images, and it's different from dataset 1 in terms of scanner manufacturers and time range. This one was collected between 2018 and 2023. Dataset 1, I think, was 2002 to 2018, if I'm not mistaken. And the table there just shows the distribution of the data points that have the mutation between the 2 datasets.
What is FLAIR scan?
FLAIR is it's a type of MRI scan that usually has things like figures. In my experience. A lot of times you see T2 and FLAIR scan combined, so they're very close to each other. Okay, so the first stage is training a CL network on paired MR images and radiology reports. It's a classification task, so if the report and the image match, it's labeled as 1. If they don't match, it's labeled as 0. This figure here shows the complete framework of the proposed method. The goal is to align both the global and local representations of the images and reports with each other. And we will get into the losses that enable that in a bit, but for now, let's focus on how the representations of each modality were acquired. The MR images were encoded using a 3D ResNet, initialized with medical net weights. It has 4 convolutional modules and a self-attention layer at the end. The first 2 modules are frozen, and the last 2 are fine-tuned to help stabilize performance and also reduce the number of weighted parameters required. The global MRI representation is the extracted output of the self-attention layer. This is kind of shown in the top row of the figure, right here. And this is an example of a radiology report. It contains description of, it's kind of hard to read, but it contains descriptions of relevant regions and important information. For example, I'll read one of the lines. Evaluation of the brain demonstrates a prominent bilobed right and especially left-sided paramedical extraaxial mass centered overlaying the posterior, frontal, and anterior parietal lobes. Or the nasal cavity appears unremarkable. So this is kind of just an idea of what's inside these reports, in case you haven't seen them before, because I didn't know what exactly was in them. But to turn these reports into useful text representations, the authors use Longformer, which is a language transformer. And it accepts input sequences of length up to 4,096 tokens, which is good for long text like radiology reports. The Longformer model was initialized with clinical Longformer weights, which is from a version of Longformer trained on MIMIC3, a large data set containing health-related information about patients admitted to critical care units. Like the ResNet model for the image representations, the first 10 layers here are frozen and the last 2 are fine-tuned. This outputs 2 types of embeddings. There is the classify or the CLS token embedding, which is a special token used by the transformer as an aggregation of the representation of the entire text. And there are just the main text embeddings. The CLS token is used for the global report representation, and the main embeddings are used for the local report representation. So any questions here before I move on? So global, local, let's start with the global representations and the global loss. The big goal of this loss is to bring the global representations of MRI images that we got from the ResNet and their corresponding radiology reports from the CLS token closer together. And they do this using a modified triplet loss. So going back to Karen's description, the aim here is to minimize the distance between similar global image and report embeddings and maximize distance between unmatched ones. So right here, given an anchor, say in this case an MR image, we are trying to minimize the distance to its positive match, which is going to be its corresponding report, and at the same time maximize its distance between the image and an unmatched report. All 3 of these elements, the anchor, the positive, and the negative, are based on data batches. Specifying the anchor and the positive is straightforward because they're paired together in the batches already. But there are different techniques for selecting the negative representation, and they've listed them here. There is random negative sampling, in which you just randomly select a disconnected sample as the negative, but that might not lead to the best ideal representations. There is hard negative sampling, where you measure the distance from every single disconnected sample to the anchor and pick the one with the lowest distance to the anchor. So AKA, the most difficult sample to be identified as a negative is used as the negative representation, so the model learns from difficult cases. But this takes a lot of time because you have to calculate the distance for all 15 disconnected samples in a batch for every single anchor in the batch, so it takes a long time to calculate. The authors instead introduce semi-hard negative sampling, which is kind of a combination of the previous 2. They sample 2 random negative representations, And they pick the one with the lower distance to the anchor, and they use that one. Yeah.
So they're trying to classify, is it like a, how many classes? Two? They're classifying between the two different, like, mutations?
No, we're not there yet. So the mutations come in later. This is just how the contrastive learning selects what the negative sample is going to be, or the mismatch sample. And there are different ways of selecting, and they all have negatives. Does that make sense? Yes. Okay. So we randomly pick two negative or mismatched representations, pick the one with the lowest distance to the anchor between those two, and select that as your negative sample. And I've included the equation here, but I'll go into more detail in a second. Okay. Tumor location is also another important part of consideration because it's an important factor in different brain tumor diagnostic tasks. So to improve the CL framework that they've come up with so far and the representation learning process, tumor location is incorporated as an external variable. In the cases where an MR image is not matched with a report, but they correspond to the same tumor location, that distance is less than an unmatched pair that doesn't have the same tumor location. So this is represented in the loss as a coefficient in the range 0 to 1. In the range 0 to 1, and it's applied to unmatched pairs with the same tumor location. So here on the right, I've created a little figure which helps you see. So saying that the anchor is an MR image right here, the distance to the matched pair is minimized, and the distance to the unmatched reports are maximized. However, if one report has the same tumor location as the MR image, that distance is going to be less compared to the report that doesn't have that matched tumor. Okay? So actually looking at the equation now, the outer summation term just specifies whether the anchor is an image, value 1, or a report, value 2 in this case. N in the second summation, or the inner summation, is the batch size, 16 here in this case. The term Aij represents the J of anchor in the batch. Pij is a positive sample that is matched with that anchor, and Nij is a negative sample that is not matched with the anchor. L refers to tumor location. It is set to 1 if it is a match, it's set to 0 otherwise. C is the tumor penalty, so it's only applied if the tumor location is a match and the value that the authors chose for it is 0.5. And the margin M is set to 0.25 in this case. So just to summarize, the first term here calculates the distance from the anchor to the positive. The second term calculates the distance from the anchor to the negative if the tumors don't match. And the third term calculates the distance from the anchor to the negative if the tumors do match. And this full measurement so the loss takes the maximum of 0 or this full measurement. The anchor should be closer to the positive than to the negative by at least margin M. And if that's already true, the measurement is negative, and so the loss gets set to 0. Otherwise, it's positive and it causes an update. Does that make sense? Okay. So that's for the global CL loss. Now there's also a local CL loss. The goal of the local loss is to align specific parts of the MR images with specific parts of the radiology report. For example, if the report says there is a lesion in the thalamus, the local loss is designed to align the local image patch of the thalamus with corresponding words or phrases in the text. And to achieve this cross-modal relevance, the authors use both the original embeddings or tokens and also weighted local embeddings, which are cross-attention weighted representations from the other modality. The MRI embeddings are captured from the output of the third convolutional module in the ResNet that they trained earlier. The reason why was this layer is typically fine-grained and has low-level visual features, which they thought was helpful. And the Report embeddings are the embedding vectors of all non-CLS tokens in the text, so that other all main one that we found earlier. Cross-attention is used to capture correlations between the local image and text representations and get a weighted local representation of each. So for example, an MRI to report cross-attention would take each local image patch as a query and append it to all the words in the report to get a weighted MRI representation, and then vice versa for the report to MRI. These are then used in another triplet loss. Very similar to the other one, the outer summation just determines whether it's a local MRI one or report representation. The second summation determines whether it's original or weighted, and then the rest is exactly the same. AIJK is the jth weighted or original local representation in the batch to the anchor. P whatever is the original weighted local representation matched with the anchor, the positive, and NKIJ is the original or weighted local representation disconnected with the anchor. So that's the local CF loss. And then these two are combined together to create a total CF loss, which is alpha times the global loss plus beta times the global loss plus beta times the local loss, where alpha and beta are trainable parameters. And I've included this figure back in here to just combine everything we just learned together. Does that make sense? Are there any questions at this point? Okay, so now that the contrastive learning framework is trained, it's time to apply the learned MRI representations to the downstream PLGG genetic marker classification. So a 3D ResNet is initialized with the weights from the CL framework we just trained. The first two convolutional models are frozen and the last two are fine-tuned. And there is a fully connected layer to perform the classification of the image into BRAF fusion or BRAF mutation. It is trained using cross-entropy loss. The classifier is trained on the 204 image subsets from dataset 1 that I pointed out earlier. And they say that this works because the pre-trained MRI encoder already understands what features radiologists consider important via the contrastive learning alignment with the reports that we did earlier. So now it serves as a better initialization for classification. They also visualize the attention map of the model to compare it against the ground truth mask to evaluate explainability of the model. These are the downstream classification performance evaluation results. 3 models are considered 2 baselines and then the one that they came up with. The 2 baselines are a 3D ResNet model that is trained entirely from scratch. The other is a 3D ResNet that is initialized with medical net weights and fine-tuned on the downstream classification labels. And their model is a 3D ResNet pre-trained using the CL framework and fine-tuned on the classification.
Slow down.
What?
Yeah, just slow down here.
Okay.
I'm a bit confused on like what the task is. Like, what are they classifying?
They're trying to classify, from what I understand, whether it is a BRAF fusion or a BRAF mutation.
Okay.
From these two right here.
Have they tried what is, without fine tuning, what is the accuracy of their pre-trained contrastive model?
I don't think they give results without the fine tuning.
Oh, I think they had the 3D ResNet trained from scratch. Oh, okay, that's not their model.
It's not their model.
Yeah. We need that's weird. So yeah, it's very hard with those results to verify them. It would be good if they took their model and trained it from scratch in supervised fashion, just supervised, right? Because we often see that you can get as high, if your goal is just classification, you can get as high or higher than self-supervised pre-trained model. Or this fine-tuning, it's kind of undoes whatever the contrastive learning is doing, because those are different goals.
Yeah.
The goal of contrastive learning is more about robustness, and you suffer because of robustness. And so when I see the numbers, when it's good, like better everywhere, well, it just doesn't add up, because contrastive learning makes the model more robust at the cost of being less precise in a sense. And they're saying precision doesn't suffer, precision improve. Recall suffers, but that means robustness. Yeah, I would be maybe Alex kind of trained me on that, but I would be much more detailed on like kind of experiments here. I believe for narrow domains, supervised training is still better.
Okay.
But that's not the point. That's okay. It's like I guess to publish a paper, yes, you need to convince reviewers, so we better everywhere. But it just doesn't make sense. And I don't know. That's what I was trying to. What did they do? And MedicalNet randomly initialized. But I think the architecture is the same. You said it's a different model, but it should be the same model, no?
It's the same model, like I guess
Just differently
different
initialized.
weights, basically. Initialized with the different weights.
Well, but then maybe they're just not training it long enough, because I don't understand where the learning curve. So if it's the same model, different initialization, are we sure that we're getting into like a complete plateau, like the weights determine it that much? I don't think the weights could determine if you train longer. It should get to the same point practically. I don't know. But the point would be faster or less, or slower. With some initializations, it should get to higher numbers quicker. That's fine. But with others, longer training. Okay, thanks. I'm done.
And so these are the mean test classification performance results on dataset 1 and dataset 2. In dataset 2, the proposed model outperforms the other two by quite a big margin. In the dataset 1, it's better in the AUC precision and F1 score, but not so great in the recall. And so they say that loading the proposed CL weights into the 3D resident model significantly enhances the performance across all the different metrics that they test. They also do some explainability evaluation. So to evaluate it, they classification models attention heat maps are visualized based on the weights extracted from the self-attention module and compared with the ground truth tumor segmentation masks. The attention heat maps are normalized in the range 0 to 1 and thresholded at 0.01 to create a binary mask. Greater overlap between them means that the model has focused on the relative parts of the image where the biopsy was taken to get those genetic markers, which means greater explainability. This table shows the 3D and 2D dye scores for this overlap, showing that the proposed CL model achieves the highest scores out of the 3 models tested, and 2D dice was based on the MRI slice that had the largest cross-section with the tumor segmentation. Yeah. This is on dataset 1, and I'll show results for dataset 2. This is kind of what I was talking about with the attention heat maps. This is with the randomly initialized model with MedicalNet, and then with the Bear model. And in this example, the attention heat map is right where the biopsy was taken and where the tumor is, whereas the other ones are in random locations. This is testing on dataset 2. Here they included 2 additional state-of-the-art methods to visualize the attention maps of the randomly initialized model, because they were like, maybe that way isn't good. They used this GradCam method and the saliency method, and still their method, the proposed CL, is performing much greater than the others. And they say that these findings, specifically with the external results, demonstrate that their framework is capable of dealing with various data shifts, including those caused by chronological and scanner variations. And the baseline models are completely unable to generalize to this second external data sets where they rely on out tumor diagnosis shown here. Yeah, the pre-training stage enables the downstream model to identify relevant image areas, aligning its attention maps with clinical evidence. And then kind of the last thing that I have to show are these ablation studies. So they do 5 ablation experiments. In the first 3, they train the framework with global loss only, with the local loss only, and with both the global and local loss without adding that tumor location coefficient. And their other scores are better than these, basically. In the experiment 4, they replaced triplet loss with contrastive loss. And in experiment 5, they used hard negative sampling for the negative sample selection instead of the semi-hard that they came up with. And yeah, across all of these, the mean AUC is highest in the proposed method. And the only difference is in the contrastive loss, the mean F1 score is greater than in the proposed method. And I think they attribute part of this to the fact their data set size is so small, because that's the only data they had access to for this classification test. But yeah, that's it. Do you have any opinions? I know you said you read the paper.
Yeah, I have a lot. They said they only sampled from one hospital as well.
Yeah, it's their own hospital.
They did show the supplemental information. Would you, if you would rerun it, do you think you'd have similar results?
Similar results?
Yeah. Do you think you'd have? Because I think that their whole entire point was that they're redoing away with this whole system because of the lack of explainability.
Yeah.
And and using this as the system and claiming that it performs even better than the previous one. So if you think you would rerun it, do you think you get the same results?
I would like. I'm a little bit. I haven't read any other papers about this specific classification. Their scores, I don't know if these are considered low for this task or if they're the highest for this task, but like 76 seems kind of low, and this die score between the attention maps and the manual tumor segmentation, like 30 is kind of low, but I don't know if that's necessarily means like it doesn't perform that well or if it is kind of the best that they have so far. I really like this kind of thing they do. I don't know if I'd have to experiment with it, but I like this attention visualization thing that they do, which is cool. I feel like I get similar results, but I have to see.
One other question: why did you choose this paper? That was really cool, but I was just curious why you chose it.
I don't have a good reason. I just saw it and I thought it was interesting, and I didn't know much about contrastive learning, so I was like, okay. Good.
But really, what they're doing is metric learning, and they're using metric loss and everything. It's kind of falls into self-supervised learning, but contrastive, usually it's not contrastive, noise contrastive. It's a little different. Like, I know that different branding, different kind of fields, they name things for various sociological reasons. But yeah, the metric learning was their first, was the same exact thing. The triplet loss is a link from metric learning. It's tiny cliqueish subcommunity. But yeah, it's kind of a natural, in retrospect, of course, natural way of let's just make it pool. Because remember the papers that we looked at, the Platonic hypothesis and things like that, like where the concepts that are semantically close are pulled geometrically together.
Mm-hmm.
So that comes from those ideas. It's like things that are similar in our head should be similar geometrically in the representations where the network maps it, and things that are dissimilar in our head according to our knowledge. We know we choose it somehow, right? Should be dissimilar or should be further away. So, but those are standard ideas from optimization theories and stuff that push the dissimilar stuff further away. It's like Tisney, and methods like that. What are they called? The nonlinear dimensionality reduction, the embeddings and stuff. They also work on the same similar principles. But there is noise contrastive estimation that is different. It's where you kind of put the probability, uniform probability or noisy probability, and the actual data probability, and maximize scale divergence between those, the minimize scale divergence, or you want to push it away. So those are closer to independence or mutual information kind of metrics. So I don't know, just a comment, basically, what people call stuff. And also, this picture, hard to trust. It's more of a cartoon.
Yeah, but it's a cool picture.
Yeah, I mean, it's a cartoon. I don't believe that random is like that. I don't believe that medical net is like that either. And then, yeah, I never seen models be that wrong on this stuff. Yeah, I mean, there may be a little off. Yeah. Focusing away from the brain.
Yeah.
No, it's a cartoon. They made it so like to get the point. Other models are bad. That's a good point to make. Yeah. So that's very interesting.
I really a couple things I liked about this paper. To play devil's advocate, your devil's advocate, is I really did like how they clearly defined the limitations of their study. I think they even clearly stated that it would work. We didn't look into the types of cancers, the types of tumors. I think it was, which is cool. I like that they clearly stated that we think a large sample size, even though we think it perform better. So I like that they understood the limitations. Comment, but yeah.
Yeah. That's good. I'm just trying to be naturally cynical about anything. Just what could go wrong or what is like. But that's not okay. My largest concern is sociological in a sense that when a new cool idea that is collectively perceived as cool excites the medium, the human goo, it starts bubbling and everyone is like, yeah, and there is no time yet to check this stuff, and it feels like some of the beliefs get propagated into the papers without being checked. And again, thanks to Alex being very OCD kind of type, when checked everything, we're kind of like empirically, we've done lots and lots, tons of unjustifiably many experiments that he shouldn't have done. He was deliberately trying to kill his defense, his dissertation, basically. It's like he was working against himself. And like, no, like supervised learning is still the best way to, like a scalpel, like the Lancet one. If you have a goal of classification, you'll get it as high as. This is the contrastive learning has a different role, but the belief is, oh, this is a cool new shiny. It should be good everywhere, and like, yeah, classification should be better than this and this and that. And so I'm naturally skeptical about this things. Gotta be careful. And back to Richard Feynman, who said he believed all the kind of professional people until one of the painters started telling him that he can get any color by mixing the primary colors and stuff. And he's like, how do you get pink? Like, oh, blah blah blah. He told him the mixture, and Feynman was like, okay. People just do their craft. They don't know what they're doing sometimes. Not this particular paper, but often. And so when I read the paper, and I did the craft myself, and like I know, like no, like I can like often when it's like if I have done it and I know what the community thinks of it, I can I can separate, like, okay, they're writing this either because they don't know but they only heard what the community is doing, or because they know and cynically writing catering to the community. It's like, because if you just try it yourself, you see it's not the case. But because as a community we tend to stand on the shoulders of each other, like a tower of lemmings, right? So yeah, uh, yeah, that's my visual, and that's like, some of the statements are wrong. It's we were just like, just there is no time. Like, hey, yeah, there is a paper that says that. Let's just do it.
I wonder if they're gonna write another grant for a paper down the line saying, you know, we did this, but we now maybe we need a bigger sample size so they can draw it out for asking for more money. I was wondering about if they'd ever do that.
You've gotten too cynical for this.
Now I could go
No, like yeah, it's natural. It's just like that's why don't believe everything as scientists. You cannot believe everything that's written, and especially like, when you've tried things yourself and you see that that's not the case, and you can like show just one single thing is not a proof, but if you have experience and then you start thinking like with the machine, it took us years to figure out like it's not what people are saying, and it's like working and blah 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 stuff like that. Yeah, yeah. Don't trust anything what people say, even in reviewed papers. Just always turn on whatever system one or system two, but the slow, painful, unforgiving, and thankless. Yeah. If you, I mean, oh no, oh don't because otherwise it's just propaganda. It's like, yeah. Okay, yeah, thanks. That's nice choice, and I hope you learn about contrastive learning, which is metric learning in this case, but that's fine. Contrastive learning is, as I said,
it's contrastive learning. It's contrasting.
in contrast to, I mean, yeah. Well, but there is actual contrastive learning where this stuff is contrasted. As yeah, but well, I guess metric. You can think of it as contrastive. Learn to come up with good names that people like, and then that's part. Well, because 99% of the science is language. When Leibniz made the language of calculus in 1670 something, it calculus totally became everyone's tool because the language was. We still use his language. We still use exactly he spent a lot of time. You can go to Munich to Leibniz's museum and see a lot of his notes where he's trying what will be a symbol for integral, what will be this and stuff like that. So everywhere in science, when we have common language, because sorry, humans are dumb. It's the societies are smart, but for society to function and focus on something, we need the common signal messaging system, which is language. So when we come up with the names that everyone understands and that kind of describe, because some people publish papers way earlier than the other communities, but they call things differently. Something that people didn't. It just doesn't catch on because people don't understand what they were talking about. Blah blah blah.
My pet peeve is when different fields have the exact same word for something, but it has a completely different meaning.
Consciousness.
Yeah, variable hydrolysis. Hydrolysis apparently means something completely different in organic chemistry in comparison to geology. And I think it's because it's based on the prefix of the Greek word hydro from water. And so, yeah, going back and forth between those is so gross. Yeah, anyway, sorry.
Yeah. Well, but that's how the. Well, I mean, I guess that's good. That's how silos form, and we need membranes in order to evolve. So we don't have like complete gray goo or whatever human colors.
Just two.
Yeah. So one community develops, another community develops. Yeah, that's cool. Okay. So we're taking a break for 2 weeks, right? And then we'll resume with a planning meeting. We don't have anything planned yet, right?
So for the next 2 weeks, we don't have anything, and then I'm scheduled for the 11th.
Oh, so we have.
If you want to take it, you're welcome.
But are you the last one in the planning session?
No, and then we have Yarong, then we have Sergey on the 25th.
Oh, okay, good. So we have the whole summer plan.
Yes.
So that was originally good goal to leave those weeks on, so you can all publish your papers. The papers that are closed should be published in this 2 weeks. Yeah.
That's why did you do that? the out if you needed it, if that makes sense. So like
Okay, I'm stopping the recording. Thanks for watching us. There's got that far. I don't think anyone gets that this far on YouTube, so we can say anything here, right?